Hello friends, today we will gain knowledge about evolution of crops, genetic engineering and genetic manipulation. And the main objectives of this topic will be: first, to discuss the concept of evolution of new crops; second, to discuss the role of biotechnology in evolution of new crops; third, to familiarize students with the concept of selective cross breeding; fourth, to describe briefly the concept of classical breeding with induced mutation and to compare the classical breeding and crop breeding through genetic engineering. First of all, let's have a general introduction about evolution of crops. The origins of agriculture and domesticated crops are intertwined, and the change from a hunter-gatherer mode to tillage, sowing and harvesting was one of the major technological innovations of humankind. There is good evidence that this occurred some 10,000 years ago in several different locations and involved the domestication of wild relatives of the major crops. Domestication involves change in the genetic makeup and morphological appearance of plants. These changes occur because people select the variations of a wild plant that best suit their needs. If the desired features can be passed to offspring over generations of planting and harvesting, the strains of plants grown change. Despite the fact that these domesticated varieties of plants are preferred over their original forebears, the wild relatives of crop plants continue to be an important resource. Reserves of wild plants offer a pool of genetic diversity. Traits of plants that might have been lost in domestication sometimes become crucial for protection of domesticated crops from stress and disease. Maintaining wild strains for their gene pool helps ensure food security. Knowledge of crop origins is thus of considerable practical importance even when these original strains are no longer harvested as crops. Development today of new crops such as perennial alternatives of currently used annual staples has potential value in helping meet serious current agricultural challenges such as the need for water use efficiency, better management of land salinization and soil conservation. A Swiss botanist, Alphonse de Candolle, started studies of the origins of crops in 1885 and proposed two approaches to answer these questions. The first is identification of the geographical distribution of wild relatives of modern crops based on careful botanical descriptions and tests for cross-pollination between candidate ancestors and the crop in question. Second, archaeological studies often provide clues on how and when a transition from hunter-gatherer existence to agriculture occurred. About 10,000 years BC, people harvested their food from the natural biological diversity that surrounded them and eventually domesticated crops and animals. During the process of domestication, people began to select better plant materials for propagation and animals for breeding, initially unwittingly, but ultimately with the intention of developing improved food crops and livestock. Over thousands of years, farmers selected for desirable traits in crops and thus improved the plants for agricultural purposes. Desirable traits included crop varieties, also known as cultivars, from cultivated varieties with shortened growing seasons, increased resistance to disease and pests, larger seeds and fruits, nutritional content, shelf life, and better adaptation to diverse ecological conditions under which crops were grown. Over the centuries, agricultural technology developed a broad spectrum of options for food, feed, and fiber production. In many ways, technology reduces the amount of time we dedicate to basic activities like food production and makes our lives easier and more enjoyable. Everyone is familiar with how transportation has changed over time to be more efficient and safer. Agriculture has also undergone tremendous changes, many of which have made food and fiber production more efficient and safer. For example, in 1870, the total population of the USA was 3 crore 85 lakh 58,371 and 53% of this population was involved in farming. In 2000, the total population was 27 crore 5 lakh and only 1.8% of the population was involved in farming. There are negative aspects to having so few members of society involved in agriculture, but this serves to illustrate how technological developments have reduced the need for basic farm labor. Agriculturists started conducting selective breeding of crops before having a thorough understanding of the basis of genetics. Gregor Mendel's discoveries explaining how traits pass from parents to offspring shed new light on the matter. Mendel's work showed that genes separate during the formation of gametes and unite randomly during fertilization. He also showed that genes are transmitted independently of one another to offspring. This understanding of the way that plants and animals acquire traits from parents created the potential for people to selectively breed crops and livestock. Gregor Mendel's discovery revolutionized agriculture by launching the development of selective crossbreeding with a comprehensive understanding of the underlying mechanisms of inheritance. After first objective, dear students, we will discuss our second objective of today's topic, that is, role of biotechnology in evolution of new crops. The basic structure of DNA is identical in all living things. In all organisms, different characteristics are determined by the sequence of the DNA base pairs. Biotechnology has developed to the point where researchers can take one or more specific genes from nearly any organism, including plants, animals, bacteria or viruses, and introduce those genes into the genome of another organism. This is called recombinant DNA technology. In agricultural biotechnology, changes are made directly to the plant's genome. Once the gene that determines a desirable trait is identified, it can be selected, extracted, and transferred directly into another plant genome. Plants that have genes from other organisms are referred to as transgenic. The presence of the desired gene controlling the trait can be tested for at any stage of growth, such as in small seedlings in a greenhouse tray. A breeder can thus quickly evaluate the plants that are produced and then select those that best express the desired trait. Producing new varieties of crops through genetic engineering takes about 10 years on average. The applications of genetic engineering through recombinant DNA technology increased with time, and the first small-scale field trials of genetically engineered plant varieties were planted in the USA and Canada in 1990, followed by the first commercial release of genetically engineered crops in 1992. Since that time, adoption of genetic engineered plants by farmers has increased annually. While the benefits of genetically engineered crop varieties have been widely recognized, there has been extensive opposition to this technology from environmental perspectives because of ethics considerations and people concerned with corporate control of crop varieties. The rise of commercialized genetically modified crops has provided economic benefit to farmers in many different countries, but has also been the source of most of the controversy surrounding the technology. This has been present since its early use. The first field trials were destroyed by anti-GM activists. Although there is a scientific consensus that currently available food derived from GM crops poses no greater risk to human health than conventional food, GM food safety is a leading concern with critics. Gene flow, impact on non-target organisms, control of the food supply, and intellectual property rights have also been raised as potential issues. These concerns have led to the development of a regulatory framework which started in 1975. It has led to an international treaty, the Cartagena Protocol on Biosafety, that was adopted in 2000. Individual countries have developed their own regulatory systems regarding GMOs, with the most marked differences occurring between the USA and Europe. New traits introduced to crop plants by genetic engineering have the potential to increase crop yields, improve agricultural practices, or add nutritional quality to products. For example, transgenic crop plants capable of degrading weed killers allow farmers to spray weeds without affecting yield. Use of herbicide-tolerant crops may also allow farmers to move away from pre-emergent herbicides and reduce tillage, thereby decreasing soil erosion and water loss. Transgenic plants that express insecticidal toxins resist attacks from insects. Crops engineered to resist insects are an alternative to sprays which may not reach all parts of the plant. They are also cost effective, reducing the use of synthetic insecticides. Genetic engineering has also been used to increase the nutritional value of food. For example, golden rice is engineered to produce beta-carotene. Edible vaccines present in the plants we eat may be on the horizon. The new traits expressed in such transgenic plants are derived from a variety of other organisms. Scientists have given a gene from the bacterium Salmonella to cultivars of soya bean, corn, canola and cotton to degrade the pesticide glyphosate. The gene for the insecticidal toxin in transgenic cotton, potato and corn plants comes from the bacterium Bacillus thuringiensis. One of the genes allowing vitamin A production in golden rice is derived from the bacterium Erwinia iridivora, others from the daffodil. The development of golden rice involved the introduction of several genes into a plant to provide a multi-step biochemical pathway. Rice grain, which serves as a food staple for much of the world, lacks vitamin A. An estimated 100 million to 200 million children worldwide have vitamin A deficiency, a condition that causes blindness and increases susceptibility to diarrhea, respiratory infection and childhood diseases such as measles. Beta-carotene and other carotenes, the red, yellow and orange pigments found in carrots and other vegetables are the precursors of vitamin A. Rice synthesizes beta-carotene in its chloroplasts but not in the edible seed tissue. Ingo Potricus and his colleagues found that geranyl diphosphate, a precursor to carotenoid production, is present in rice seed. They genetically engineered golden rice to express the enzymes necessary for the conversion of geranyl diphosphate to beta-carotene. The synthesis of beta-carotene from geranyl diphosphate requires 4 biochemical reactions, each catalyzed by a different enzyme. A bacterium, Agrobacterium tumefaciens, containing 3 plasmids, was used to introduce all the genes necessary for the complete biochemical pathway for beta-carotene production. It was possible to use 3 enzymes instead of 4 because the bacterial enzyme phytoene desaturase accomplishes what 2 plant enzymes phytoene desaturase and beta-carotene desaturase do. Edible vaccines available in crops could help people in developing nations where transportation, refrigeration and disposable needle supplies are limited. Hugh Mason and his colleagues have expressed a gene that encodes an E. coli protein in potatoes. Volunteers who ate raw, modified potatoes developed antibodies to the protein. Research is underway to see whether the antibodies will protect against diarrhea induced by disease causing E. coli. The first genetically modified crop plant was produced in 1982, an antibiotic resistance tobacco plant. The first field trials occurred in France and the USA in 1986 when tobacco plants were engineered for herbicide resistance. In 1987, Plant Genetic Systems, founded by Mark Van Montague and Jeff Shell, was the first company to genetically engineer insect-resistant plants by incorporating genes that produced insecticidal proteins from Bacillus thuringiensis. The People's Republic of China was the first country to allow commercialized transgenic plants, introduced a virus-resistant tobacco in 1992, which was withdrawn in 1997. The first genetically modified crop approved for sale in the US in 1994 was the Flavor Saver tomato. It had a longer shelf life because it took longer to soften after ripening. In 1994, the European Union approved tobacco engineered to be resistant to the herbicide bromoxynil, making it the first commercially genetically engineered crop marketed in Europe. In 1995, Bt potato was approved by the US Environmental Protection Agency, making it the country's first pesticide-producing crop. In 1995, canola with modified oil composition, Bt maize, bromoxynil-resistant cotton, Bt cotton, glyphosate-resistant soya beans, virus-resistant squash, and additional delayed ripening tomatoes were approved. As of mid-1996, a total of 35 approvals had been granted to commercially grow 8 transgenic crops and 1 flower crop, with 8 different traits in 6 countries plus the EU. In 2000, vitamin A enriched golden rice was developed, though as of 2016 it was not yet in commercial production. In 2013, the leaders of the 3 research teams that first applied genetic engineering to crops, Robert Fraley, Mark Van Montague, and Mary Dell Chilton, were awarded the World Food Prize for improving the quality, quantity, or availability of food in the world. Dear students, now we will discuss third objective, that is selective crossbreeding. In traditional plant breeding, new varieties are developed either by selecting plants with desirable characteristics or by combining qualities from 2 closely related plants through selective breeding. These features may, for example, be resistance to a particular pest or disease or tolerance to climatic conditions. Traditional plant breeding has produced numerous highly successful new varieties of crops over the centuries. There have also been many less than successful crosses made. In traditional breeding, crosses are often made in a relatively uncontrolled manner. The breeder chooses the parents to cross, but at the genetic level, the results are unpredictable. DNA from the parents recombines randomly, and desirable traits such as pest resistance may be bundled with undesirable traits such as lower yield or poor quality. The parent plants must be closely related to produce offspring. Traditional breeding programs are time-consuming, often taking decades to produce new viable crop varieties, and labor-intensive. A great deal of effort is required to separate undesirable from desirable traits, and this is not always economically practical. Many potential benefits are lost along the way as plants that fail to demonstrate the introduced characteristics are discarded. Traditional plant breeding takes on average 12 to 15 years to produce a new crop variety. Dear students, after discussing third objective, we will discuss our fourth objective of today's topic, that is classical breeding with induced mutations. Mutations are changes in the genetic makeup of a plant. Mutations occur naturally and sometimes result in the development of new beneficial traits. In 1940, plant breeders learned that they could make mutations happen faster with a process called mutagenesis. Radiation or chemicals are used to change the plant's DNA, the basic molecular system of all organisms' genetic material. The goal is to cause changes in the sequence of the base pairs of DNA, which provide biochemical instructions for the development of plants. Resultant plants may possess new and desirable characteristics through this modification of their genetic material. During this process, plant breeders must grow and evaluate each plant from each seed produced. More than 2,500 plant varieties, including rice, wheat, grapefruit, lettuce, and many fruits, have been developed using radiation mutagenesis. Induced mutation breeding was widely used in the United States during 1970s, but today few varieties are produced using this technique. As our understanding of genetics developed, so new technologies for plant variety development arose. Examples of these that are used today include genetic marker assisted breeding, where molecular markers associated with specific traits could be used to direct breeding programs, and genetic engineering. Some of the significant steps leading to the current state of the art are explained. First, discovery by Watson and Crick, that is structure of DNA in 1953. Another milestone in the development of understanding of genetics and how genes function was the discovery of the structure of DNA and how DNA works. Two scientists, James Watson and Francis Crick, made this discovery considered to be one of the most significant scientific works in biology, largely through synthesis of the work of other scientists. Their work contributed significantly to understanding what genes were. Second, discovering genes that move, that is, transposons. Transposons are sections of DNA genes that move from one location to another on a chromosome. Transposons have been referred to as jumping genes, genes that are able to move around. Interestingly, transposons may be manipulated to alter the DNA inside living organisms. Barbara McClinton discovered an interesting effect of transposons. She was able to show how the changes in DNA caused by transposons affected the color of maize kernels. Third, tissue culture and plant regeneration. Another significant development in technology that was important for plant breeding was the development of micropropagation techniques known as tissue culture. Tissue culture permits researchers to clone plant material by excising small amounts of tissue from plants of interest and then inducing growth of the tissue on media to ultimately form a new plant. This new plant carries the entire genetic information of the donor plant. Exact copies of a desired plant could thus be produced without depending on pollinators, the need for seeds, and this could all be done quickly. Fourth is embryo rescue. Often when distantly related plant species are hybridized, the embryos formed following fertilization will be aborted. The development of embryo rescue technology permitted crop breeders to make crosses among distantly related varieties and then to save the resulting embryos and then grow them into whole plants through tissue culture. Fifth is protoplast fusion. Protoplasts are cells that have lost their cell walls. The cell wall can be removed either by mechanical means or by the action of enzymes. They are left with only a cell membrane surrounding the cell. Protoplasts can be manipulated in many ways that can be used in plant breeding. This includes producing hybrid cells and using protoplasts to introduce new genes into plant cells which can then be grown using tissue culture technique. Sixth is genetic engineering. Building on the above discoveries into the 1980s, advances in the field of molecular biology provided scientists with the potential to purposely transfer DNA between organisms, whether closely or distantly related. This set the stage for potentially extremely beneficial advancement in crop breeding, but has also been very controversial. Dear students, we will discuss last objective of our today's topic, that is comparing classical breeding and crop breeding through genetic engineering. Crops produced through genetic engineering are sometimes referred to as genetically modified organisms. The term genetic modification and so-called genetically modified organisms is frequently misused. All types, that's organic, conventional of agriculture, modify the genes of plants so that they will have desirable traits. The difference is that traditional forms of breeding change the plant's genetics indirectly by selecting plants with specific traits, while genetic engineering changes the traits by making changes directly to the DNA. In traditional breeding, crosses are made in a relatively uncontrolled manner. The breeder chooses the parents to cross, but at the genetic level, the results are unpredictable. DNA from the parents recombines randomly. In contrast, genetic engineering permits highly targeted transfer of genes, quick and efficient tracking of genes in new varieties, and ultimately increased efficiency in developing new crop varieties with new and desirable traits. Dear friends, this brings us to the end of this lecture on evolution of crops, genetic engineering and genetic manipulation. Hope you have enjoyed this lecture. Have a nice day.